Hello, good morning, everyone. Thank you for being on time. We're going to go ahead and get started. Let's begin by finishing up the cardiovascular system. We still have a couple of slides from last time that talks about a few cardiovascular diseases. So here we go. Now, you probably have heard of cholesterol, right? Cholesterol is something that you don't want too much of in your body, but it's necessary to have cholesterol for things like, cell membrane. We learned last semester the cholesterol helps maintain the fluidity in the cell membrane. We also need cholesterol to use as a raw material for making things like testosterone, progesterone. So, it's not something that we want to keep completely out of the body, but there are good cholesterol and then there are not so good cholesterol. The thing about cholesterol is when we consume it in our diet, because it is a lipid, is not going to be able to go into the bloodstream directly. So, as we will learn later in the course, in the digestive system, there are special pathways for absorbing the cholesterol in the intestines, and it is actually transported to the liver where it gets combined with these things called lipoproteins before they can go back into the bloodstream. Depending on how much lipoproteins are incorporated into the cholesterol, we end up having what's called LDLs, which is low density lipoproteins. These are the bad ones, okay? These are the bad ones. You don't want too much of these things, okay? It causes buildup in the endothelial linings of the arteries. That's just like the innermost layer of the blood vessels, and then they could cause hardening of arteries and so on and so forth. The HDL, that's the good cholesterol. Okay, so when your doctor tests your blood, they check for the ratio between the HDL and the LDL, and then there are some ranges that would tell you that you're within a healthy, acceptable, good range. Otherwise, you might have to cut down your cholesterol, and if it's too high, you might have to take medications to control it altogether. Okay, so that's LDL and HDL. Now, as I was saying, the buildup of these fatty material, and it doesn't have to be just cholesterol, there are other fatty stuff that could build up in the arterial wall. It leads to the hardening and the scarring of the blood vessels, and that is called atherosclerosis. Okay, atherosclerosis, the scarring of these blood vessels. And what happens is it only takes a little bit of buildup to trigger sort of like a snowball effect. And here is why. I told you this last week that blood likes to pass through a smooth interior surface, right? Anytime there is a rough patch on the inside of the blood vessel, let's say there's a little bit of buildup here, right, of fatty material, then as the blood passes by, they will stick onto this part. Okay? Part of the blood will stick to it, the platelet, as we will learn, and then this thing will get bigger and bigger and bigger, and that would in turn trigger the formation of a blood clot on top of this buildup, right? And eventually, it's going to get really, really narrow. And depending on, where this is happening in the body, there could be various consequences. But let me show you a picture of what it looks like first. So over here on the right side, on the left picture of the right side here, we have a blood vessels that has been cut, right? Basically like the toilet paper that I love using. The inside here is called the lumen. Right? That's where the blood passes through. So in in a healthy blood vessels, the lumen, the inside, right? Lumen is Lumen. We learned that last time. Okay, the lumen is unobstructed. Right? It's clear, and things can pass through. Blood can pass through easily. However, as you can see on this artery, we have the atherosclerotic plaque. That's what this is, right? The name is right here, atherosclerotic plaque. That's the fatty stuff that has been building up. Yes, that's clogging the artery, but what's really making the artery very, very narrow is the blood clot that forms on top of the atherosclerotic plaque. Okay, so the fatty substance is just the start of this process. It's your body's own blood clotting on top of the plaque that causes the rest of the narrowing. And then, as you can imagine, right, if your blood vessel, go from this to this, then it's substantially much narrower, and the blood is going to have a much harder time going through. And like I said, depending on where in the body this happens, the consequences could be very different. For example, if it happens in the coronary artery, okay, then the person is going to potentially have a heart attack. So way back at the beginning of a lecture, right, there's a table that tells you what blood vessels supplies to different parts of the body, right? Pulmonary goes to the lung, carotid goes to the head, and then the heart relies on its own network of blood vessels for oxygen. Okay? The blood that is passing through the chambers, like going through the atrium and the ventricles, they are going by way too fast, and the pressure is too high. Therefore, the heart cannot use those blood. Okay? The heart muscle cannot extract oxygen and nutrients from the blood that's passing through its own chambers because it's too fast and the pressure is too high. So instead, it is going to be relying on this network of coronary arteries that are like on the surface of the heart, and then they kind of grows into the heart muscle, right? So the coronary arteries. Now, if the fatty stuff starts to form inside one of these coronary arteries, it could lead to the formation of the blood clot, and it causes the coronary artery to become narrow, right? Now, it's not clogged yet. It's not completely blocked off. It's just narrowing. If that's the case, then the person might have what we call the coronary artery disease, okay, or CAD for short. The coronary artery disease refers to a group of disease where it's affecting the coronary artery, and one of these diseases is caused by the partial blockade of the coronary artery. Okay, so when it's partially blocked, what happens is the person is going to experience chest pain when they try to physically exert themselves. So let's say they try to lift something heavy, right? Then they feel the chest pain, because when you lift something heavy, the muscle requires extra supply of energy and oxygen, and the heart try to pump harder for that. And by doing so, the heart also requires more oxygen, right? But because the coronary arteries are partially blocked, then you're not able to supply that oxygen, and then it leads to the chest pain. Same thing as climbing up a flight of stairs. Okay, the person might find themselves short of breath, and again experience a little bit of chest pain. So these kind of chest pain we call angina. Okay, you probably have heard of that before. Okay, angina. So people who have the partially blocked coronary arteries that are not super severe yet, and they experience these chest pain, they could take aspirin on a daily basis. The aspirin is the one that the people take for headaches, but aspirin has another pharmaceutical property. It's able to make your blood thinner, okay? And so the blood is able to pass through the narrow blood vessels easier. Right? If the blood is really thick, then it has a harder time passing through, right? So by taking these blood thinner, right, it's almost like a prevention medication, right? We don't wait for the chest pain to occur. Okay, we just make sure that the blood is thinner and it's able to pass through the blood vessels, easier. But what's one of the side effects of having the blood too thin? Anybody?
You can bleed out.
Exactly, right? So that's why, like, if you go to a dentist and they ask you to fill out like a questionnaire for the first time or something, they ask you if you're taking aspirin regularly, because they have to know, right? When you do the dental work, there is quite a bit of bleeding, right? And if you're on aspirin, then the bleeding could be quite severe. So another one is called nitroglycerin. Okay, nitroglycerin. It was actually discovered as an explosive. They use this in bombs, but somehow, I don't know why, I didn't really read into it. They figure out it was also good for vasodilator. Okay, vasodilator means it opens up your blood vessel. Okay, opens up blood vessel. Okay, you probably again seen this on TV at one point or another, that there's some guy who's feeling the chest pain after someone says something very bad to them and provoke them, makes them angry, and then they feel the chest pain, and then they have to reach for the pocket to get the pills, right? That pill they're taking at that moment is not aspirin, okay? Because aspirin takes a long time to kick in, right? The thing that they're taking is the nitroglycerin. Okay, they could put it under the tongue, right? And now they, I think, even have sprays form. And as soon as it gets into your bloodstream, right, it travels throughout your body, and then it opens up the blood vessels, and then it alleviates the pain by restoring the blood flow, essentially. Any questions about aspirin and nitroglycerin? Fantastic. Now, if it's really bad, then to the point that medication is not enough to control the situation, there are surgical options. One option is what we call balloon angioplasty. Okay, angioplasty. So in this case, a catheter is inserted into the blocked artery. Usually they feed it up your femoral arteries, like from the leg, and then using ultrasound, they can guide the catheter to where the block is located. But of course, like before all this, they have to locate a clot, right? And you can do an angiogram to locate a clot. You basically have some dye that are injected into your bloodstream so that it makes your blood vessel shows up in an X-ray. So they know where the clot is, okay, before this procedure. It's not like they poke the catheter in and then try to poke around to locate the clot. They know where it is already, okay? So then they're gonna guide it to that location. They're gonna open up a balloon at the end of the catheter, and by opening the balloon, it basically props open the clot blood vessels. Okay. However, the in this procedure, restenosis is common. We talked about stenosis before, right? Stenosis is the narrowing, right, of a blood vessel. So it's not uncommon for people who underwent this procedure, the blood vessel just goes back to narrow later on. Okay, so they might have to go back for a few other angioplasty. And one way to prevent that from happening is to insert a stent. Okay, so you see this, there's like a little wire meshing right there. That's a medical stent that's going to be left inside the blood vessel, okay, to prop it open essentially. Okay, but it took a long time for people to figure this out, because you can't just put any foreign body into your own body. Your immune system is going to attack it, right? So they have to make sure that they have a material that's not going to trigger your immune system, for example. Okay, and then they leave it there to keep it open. Now, there are a couple of complications here as well. When you pop open the blood vessels, sometimes it's possible for a little bit of debris to break off from the plaque, and then it could travel downstream. If that little small plaque gets stuck in a smaller blood vessels, then it could cut off supply to that part of the body, right? So it's not without risk. Oh, I just see a message. It's the same as. Oh yeah, okay. You answer your own question. Very good. That's it also involve a catheter, but then you are putting the valve in the aorta in that case, replacing the valve in the aorta. Anyways, so that's that. So what's another option for balloon angioplasty? We have what's called coronary bypass. Okay, coronary bypass. This is when it's like completely blocked. If it's completely blocked, then we have a heart attack. Okay, you have no oxygen supplying to the part of the heart, and the a muscle starts to die off, right? And that leads to a heart attack. The proper name, the medical term for heart attack is myocardial infarction. Myocardial is referring to the myocardium, right? That's the heart muscle. And infarction just means dying. So when you hear a cold blue at the hospital, that means someone is having a heart attack. Someone's having a myocardial infarction. So if the person survives the heart attack, and there are ways that you can intervene when someone's having a heart attack. But afterwards, you want to consider doing coronary bypass. Okay, so in this case, I don't think the name is here for some reason. Okay, I'll just write out coronary bypass. Okay, that's when you take a vessel from another part of the body and you create a detour, right? Like an alternate way for the blood to flow beyond the clot location. So let's say there's a blood clot here and a blood clot like here and there. So the blood normally would come, but then it would get blocked there. So everything here is going to die off. Okay? So instead, you're going to create a blood vessel. You hook it up to the aorta, and then it allows the blood to flow past the block location, and it restores blood flow. Okay, so like, people can have multiple bypasses. Of course, not everything's going to go back to normal necessarily, but then, it's a way to extend the life of the heart and therefore of the person, right? Sometimes the only way is to get a heart transplant, but while you wait for the heart transplant, there are these interventions to kind of extend the life a little bit longer for the organ. And that is everything for lecture 1. I believe your quiz is next week. I honestly don't remember if it's Monday or Wednesday. I'll double check the calendar after class, and then I'll post the information on eCentennial for you, like how many questions, the time, a limit, and and all that stuff. Does anybody have any question about lecture 1 before I start number 2?
Are we going to have any class or anything for the quiz?
Or like a review?
Yeah.
Oh, no review. We will have a review for the test, but no review for the quiz. I'll post some materials for you to I mean the study guide, right? That's the main one for you to work through. Okay. All right.
Excuse me.
Yep.
Could you explain how nitrogen glycerin worked too faster to open the
blood vessels?
Yeah, blood vessel.
Yeah. So as soon as it gets into the bloodstream, then the blood gets carried to the heart, and it basically binds to these I think it's called beta blockers, right? Which essentially relaxes the smooth muscles that surrounds your vessels, and then essentially once they relax, it opens up. And the reason it's faster is because it's able to dissolve and get absorbed by the body much faster than aspirin can. Okay, aspirin is a pill form. You take it, it goes into your stomach, and then it takes a while for it to get absorbed, right? The main difference between how quickly it can act is how fast you can get it into your bloodstream, essentially. Okay, and but when you are having the blockade, it takes some time for you to thin your blood as well, right? Whereas opening up the blood vessel is more instantaneous. So I hope that answers your question.
Thank you.
Yeah, okay, thank you. So I'm gonna apologize to you guys. There is something wrong with one of my my tab is leaking, and I have someone to come and fix it today. So I gotta open the door for the guy when he comes. So he said he's gonna come at around 10. So I'm gonna try to teach all the way to 10, and then we're gonna take the break when I have to open the door. I know it's not ideal that to keep you guys here so long, but I gotta fix the tab. I'm so sorry. All right, so lecture number 2. Okay, this is not on the quiz. Okay, this is not on the quiz, but it's on your test. Okay, and I promise you, this is really fun. Okay, this blood stuff, I love talking about it. It's really relatable to many people, especially if you had kids. When you were pregnant, your doctor probably tests for your blood type and your husband's blood type, or your spouse's blood type, to see whether there's some kind of incompatibility between you and the baby. And we're gonna talk about all that stuff. It's a really exciting topic, and it's relatable to the first lesson. First lesson is the cardiovascular system, talking about distribution of the blood, and now we're gonna look, exactly what is in the blood. And then next week, we're going to talk about the immune system, which is also an integral part of the blood, as you will see. Okay?
Professor, I have a question.
Yeah, please.
Sorry. Are we going to get the answers for the study guide?
Yes, did I not post it already? I thought I did. Let me just double check. Can someone, does anybody know if I posted it? I think I did.
It's there. It's
Is
in
there?
the content section.
Oh, okay, yeah. But you know what? Let me just show you guys one more time. So at the end of each class, right, if you just go to the course homepage, I would say lecture archive and class notes have been updated. And then study guide answers. Now you can click on that. Yeah, I think it's there. But let me just double check. Yep, it's visible. Okay, so double check again, Mohammed. If it's still not there, let me know. I'll double check for you.
Thank you.
Yep. Okay, let's do the blood thing. Blood. Okay. There are approximately 5 liters of blood in someone who's about 70 kilogram. Of course, if you are heavier or bigger build, you would have more blood, and vice versa. And what exactly is blood? Okay. Blood is essentially a protein-rich fluid. Okay. So it's some kind of liquid that has a lot of proteins in it, and also have cellular components. Okay. So we know we have red cells, white cells, right? But there are actually different kinds of white cells, which we will look at, as well as there's some platelets and so on and so forth. The protein portion of the blood is called the plasma. Okay? Plasma. That makes up 55% of your total blood volume. The cellular components, the white cell, the red cells, and the platelets, that is what we call the formed elements. Okay? So that's about 45%. Okay? Total blood. Plasma is the protein liquid portion. And then the cells, that's the formed elements. How do we separate the blood? Once you have a blood sample, you can put it in what's called a centrifuge. Okay, centrifuge gonna spin the thing at really, really, really high speed. We talked about centrifuge last semester, right? I don't know if you remember, but there was that experiment with the virus and the bacteria, right? Anyway, so when you spin the content down, the heavier stuff goes to the bottom, and then the lighter stuff goes to the top. Right? So once you spin the stuff, the plasma, which is just liquid with dissolved proteins in it, it's going to be at the top. Okay. In a healthy person, the plasma should be clear. Okay. Clear doesn't mean no color, right? Like if you think about apple juice, it's clear, right? You can see through it, right? Okay. It's not like cloudy or anything like that. So plasma should be clear, yellowish color. And then you have the formed elements that are synced to the bottom. Okay. So here, the heaviest stuff, the heaviest cell is going to be the red cell. Red cells are the heaviest because they contain which element? Anybody want to guess? You know this. You must have heard of it before.
O2, CO2.
Okay, yeah. I'm looking for
Hemoglobin?
what is it? Sorry, Patrice.
Hemoglobin.
Okay, yes, it contains hemoglobin. And what kind of metal is in hemoglobin?
Iron.
That's right, iron.
Mhm.
Right. So iron makes it really heavy, right? That's why it's going to sink to the bottom. Okay, so that's the most of it you can see is your red cell. Okay, most of the form element is your red cell. The white cells and the platelets, they are just going to be hovering here in between the plasma and the red cells. There's this little coating, very, very thin. And it turns out the leukocytes, which is the white cell, and the platelets only makes up about 1% of your total blood volume. Okay? More on that later. This is an actual picture of what it looks like. I think it's just the way that they took the picture, okay? Like the color of the plasma is supposed to be more transparent than that. Okay, I know because I've before I was a teacher, I do research, right? I told you, and I worked at the Mount Sinai Hospital and CBS before, and one of my job at the time, it was a horrible thing, was to collect cord blood. Cord blood is the blood taken from the umbilical cord of newborn babies, but I have to get the consent of the mother to do that, right? And I have to go and ask them while they are, in labor, not like delivering, but like, when they're in pain and just waiting for a delivery. I hated that thing, that job a lot, because the husband would sometimes be like, oh, just leave us alone. And I understand, right? But it was for research purposes. I had to get the cord blood. So when I get the cord blood, I would spin it down, and then you would have the plasma on top, and then you would have the red cells at the bottom, the form elements, right? Now, did you know when you donate blood, how many of you have donated blood before? Let's do a poll and see. Just curiosity. If you have donated blood before, please choose A. If you have not, please choose B. Okay, yeah, only one person has donated blood. According to the poll, that's something that you can try, right? At least once, just to see how it's like. But when you donate blood, when they take the blood out of you, you actually have to separate the blood. Okay, so it separates into the plasma, which is what this top right bag is showing you, and then you have the red cell, RBC. Plasma. Okay. And the rules for red cell transfusion is actually different from the rules for plasma transfusion. In movies, you see someone, hook up a line from their arm directly, give blood to someone else. That's not supposed to happen, right? Like even if you guys have like matching blood type, there are still going to be issues. Okay, and it's not like you have heard people say the phrase O is universal donor, right? You probably heard that people with type O blood, they are universal donor, and I'll explain to you why that phrase is a little bit not accurate. Okay, just because you're O doesn't mean you can automatically hook your blood and give it to other people. Right? In most cases, you're gonna cause harm. So in this lecture, we're gonna understand why we have to separate the blood when we donate it, and the rules for transfusion. Okay, so plasma can be frozen and it can be stored for like up to half a year. Whereas blood products, they like the RBC rather, they are only good for a couple of weeks. Okay. All right, so let's take a closer look at the plasma. Plasma is mostly water, right? And it contains dissolved ions like the ones that we would expect to be the key electrolytes in the body, like the Na plus, chloride, right? You might have some potassium, so on and so forth. Okay, basically, next time you drink a sport drinks like a Gatorade or something, you look at the back, all those electrolytes, those are basically going to be in your blood plasma. Nutrients like glucose, right? There might be some amino acids. Okay. Waste product like CO2. There might be some urea. Urea is what's going to be found in the urine, right? It's a byproduct of protein metabolism. And you will have a lot of plasma protein. A lot of plasma proteins. Okay, so these are proteins that exist within the liquid portion of your blood. The other part of the blood is the formed element, right? And so I already told you, you have your red cell, white cells, and platelets. Okay? The red cell portion has another special name. It's called a hematocrit. Okay, hematocrit. So because there's so much of it, they just give it a special name. Like when you spin it down, that big chunk of red blob that you get at the bottom, that's the hematocrit. Right? It does not include the white cells. It does not include the platelets. White cells in general, they help defend the body against pathogens. And pathogens are living organisms that can cause diseases. Okay. Bacteria, viruses, fungus, they are considered as pathogens. Cancer is not a pathogen. Okay. Poisoning is not a pathogen. So your white cells are very good at fighting pathogens, bacteria, virus, fungus, parasites, but they're not very good at dealing with cancer. They're not very good at dealing with exposure to poison. And then finally, we have the platelets, which stops the bleeding. Okay. These are just overviews. Okay. We're going to spend time taking a closer look at them. You don't have to memorize these numbers, but it's showing you the red cells is the most common. You have 5 to 6 million per millimeter cube of blood. Okay, and then white cells and platelets. It's not as abundant, right? So let's take a closer look at the plasma protein. I'm going to give you a few examples. Okay, the most abundant means like you have a lot of it. Okay, the most abundant plasma protein is something called albumin. Albumin. Okay? Albumin is made in the liver. Okay? People who take protein supplements for bodybuilding, okay, the whey that they take, the whey, that contains albumin in it. Okay? Even some baby formula, they have albumin in it. Okay? It's made in the liver, as I said. And the main function appears to be to help maintain osmotic pressure and volume. Now, go back to the previous lecture, right? Osmotic pressure is caused by a difference in concentration between the blood and the surrounding tissue. So if you have a lot of albumin in your plasma, right, then you have more dots, so to speak, right, in your bloodstream, right? And then compared to the outside, not as much. And this difference is going to help draw fluid into the bloodstream. Right? Remember, these arrows represent osmotic pressure, counteracting what other force? Anybody remember from last class? What's the opposing force?
Blood pressure.
That's right. Blood pressure keeps on pushing things out, but these osmotic pressure, counteracts them by bringing the fluid back in. And to do that, you need to have the plasma protein, right, in the blood. So that's albumin. That's what it's for. Now, what is this picture down here, right? So this picture shows kids that are malnutritioned. Okay, maybe they are in countries where they don't have access to nutritious food. And one of the hallmarks of these kids is that they have a big tummy, right? They have a big belly. Okay, and the reason is because when they don't have good diet, they don't have enough proteins in their food. They cannot make enough proteins in the body. So the albumin content, amongst other things, is going to go down. And when they have less albumin, then what happens is the osmotic pressure is now weaker, right? You're not going to be able to retain as much fluid in the bloodstream. And so between the tug of war of blood pressure pushing things out and osmotic pressure bringing things in, the blood pressure is winning, right? And so there's a lot of fluid that's oozing out of the blood vessel. Now, when you move around your arms and then your legs, the muscle helps return some of those fluid back into the circulation. But in the abdomen, right, there's not a lot of ab movement, right? And so the muscle is not able to clear the fluid as efficiently. And so there's accumulation of fluid that has leaked out from the blood vessel in the abdominal area. So that's why people who have protein deficiency, right, they have this big tummy. Does that make any sense? Questions? Okay. Now, there are other kinds of plasma proteins. I'm just introducing them to you here, but we'll come back to it and write a more detailed notes. We have proteins that are responsible for forming a blood clot. Okay, the platelets, the cells, helps the blood clot as well, but then there are other proteins that are necessary in the process. They are called fibrinogen and prothrombin. Okay, more on that later. It's just a name for now. Don't think too much about it. Okay, fibrinogen and prothrombin. If you are low in fibrinogen and prothrombin, then your blood does not clot quick enough. So that's something they have to check before surgery. If it's determined that you have low fibrin and low prothrombin, you might have to have a plasma transfusion. Okay, because plasma is where you will find these proteins, right? Antibodies. Okay. Antibodies are necessary for immune defense. We will learn a lot more about antibodies next lecture. Do not confuse antibodies with antibiotics, right? Antibiotics are pills that you take, medication that you take to kill bacteria. Antibodies are proteins that your body creates. Okay, so let's say you have chickenpox, then your body is going to make anti-chickenpox antibodies. right? They will fight off the chickenpox virus, right? If you get COVID, then you will make anti-COVID antibodies, right? Okay. So you will make whatever antibodies. The whatever, fill in the blanks, is whatever you get infected with. Okay. You got the flu, then you need the anti-flu antibodies. That's like the simplification form of what antibodies are. Okay.
So fibrinogen is platelets.
No. Platelets are the cells. Okay. But then the fibrinogen and the prothrombin, these are proteins that will work with the cell to stop the bleeding. Okay. And we will look at how that happens later on in a few more slides. Okay. Right now, I just want to show you there are a bunch of proteins in the body, right? Most of it is albumin. It keeps the pressure and the volume in the blood. And then we have some that are necessary for blood clot. And then we have some that are necessary for fighting infections. Okay.
Excuse me, sir.
Yes, Blessing?
So how come the body doesn't make antibodies for HIV?
Yes, that is a very good question. I have a bunch of HIV slides for the next lecture. Okay, so come back next week. Okay, I promise you I'll answer that. Okay.
Okay, thank you.
Okay, where do cells come from? The blood cells. Okay, where do red cells, white cells, and platelets come from? Okay, they come from active bone marrow. Okay, if you think about the bone, the inside of a long bone, for example, is hollow. Okay, again, my favorite prop, right? Okay, the inside of a bone is hollow. Okay, but then it's filled with this stuff called bone marrow. Okay, people can order bone marrow from the restaurant, right? This apparently is a, you know, it actually tastes like nothing. Okay, but they have to season it and stuff like that. So you're basically just like slurping salt. Okay, but you know, bone marrow, the function of it is to make new cells. Okay, new blood cells. Okay, and the process of making new blood cells is called hematopoiesis, formation of new blood cells. Okay, so if I ask you on the test, which of the following is the term for making new blood cells? Hematopoiesis. Hematopoiesis. Not just red, but like all the cells. Okay, so from you have stem cells in these bone marrow. I think I talked about bone marrow previously, right? Last semester. And then you would have, they would, under the right condition, they would change into cells that would ultimately become a red cell. There are different kinds of white cells here, which we will learn all about. And then we have platelets as well. Okay, you don't have to memorize this slide. The only thing I want you to get from this picture is that blood cells, they all come from bone marrow. And the process to make it is called hematopoiesis. Okay? As a grown-up, the number of sites that are available for making new blood cells is limited. Okay, so like you have active bone marrow in your femur. Femur is like, you know, right here, that's your femur, right? Okay. You have some active bone marrow in like your pelvis bone, right? So not all bones are gonna be able to make blood cells. Okay, selected sites. Okay. So that's the overview. Okay? A lot of information. But now we're gonna go into the details. Now we're gonna spend more time on each of the cell type, and you know, hopefully it will make more sense once everything is put together. This is my blood, eh? This picture here. Last year, I during the summer, I keep on getting these hives. Okay, like it was so itchy, I couldn't sleep. It just keeps on coming. Even though I took the what do you call that stuff? The pink stuff, Benadryl. Right? Even though I take the Benadryl, like it just keeps on coming back. And then my wife, her job is to analyze blood, at the hospital. Okay, so she's like, you know what, I can take a look at your blood and see if there's something wrong with it, right? So and then she sent me this picture and she said, your blood looks fine. I don't know what's wrong with you. Just write it out. And so that's what I did. Right? I didn't do anything for another 2 weeks or so, and then it just went away. We never know what happened. Okay, sometimes people just get hives for whatever reason. But that's my blood right here. And you can see the pink circles, those are the red cells, right? What do you think these purple stuff are? These big purple circle here? There are only 2 in the whole few. Anybody want to
White
guess?
blood cells?
That's right, those are the white cells, okay? Now, all cells are supposed to be transparent under the microscope, right? They are only this color because, you have to add stains to them. Stains are like special dyes, right? Okay, so it's not surprising to see so many red cells, but so few white cells, right? It will be abnormal, for example, to have a lot of white cells. Then definitely I'm fighting something, right? Okay, because if we look back to one of the earlier slides, what percent of the blood is white cells? Less than 1%, right? Okay, whereas 45% of the blood are just red cells. So if you look at any, we call this a blood smear. Blood smear. You will see a bunch of red cells, some white cells. And if you look really closely, right, these little dots right here, right, what do you think those are? There are only 3 things in the blood that we learned so far. So what do you think those little white dots are?
Plasma?
Platelets. Yeah, platelets. Yeah, platelets, right? So all these things that you're seeing, they are the formed elements, right? Formed elements. The white cells, the red cells, and the platelets. Okay, enough about my blood. Let's talk about red blood cells in general.
Excuse me, how about plasma? The space between.
Yeah, exactly, right? So the plasma would be the liquid portion, right? But in this case, the plasma is gone. Like when you prepare the slide, you washed off all the proteins and all the liquids anyways, right? So like you won't be able to see the plasma per se, okay? But to get the plasma, you would spin the blood down, and then the whole top portion, you can just siphon it off, and then you can get a bag of plasma, like in this case. Okay, and then you can freeze it. So this is plasma taken from an AB person. All right, you wouldn't really look at the plasma necessarily under the microscope. Okay, so red blood cells, RBC for short, and the fancy word for RBC, for red blood cell, is erythrocytes. Erythrocytes. Erythro means red in Latin. Cytes is cell. Okay, for real, that's what it means. Okay, so erythrocytes. They do not have a nucleus. That's why if you look at the red cells here at the bottom right corner, they are a little bit pinched in at the center. Right? There's no nucleus, so you can pinch it in the center, and that allows the cell to have more surface area. Right? The more surface area the cell has, the more efficient it will be when it comes to gas exchange. Okay, that's after all the main job of RBC. Right? They have to be able to transport oxygen, and actually they will transport carbon dioxide as well. Okay, they take the waste back to the lungs, right? And then they take the oxygen to your tissue through the circulation that we talked about last class. The molecule that allows red cells to transport oxygen is the hemoglobin. Right? I am blocking the hemoglobin picture, but that's what it is right here. You can see there are 4 protein chains, and at the center of it, we have what's called a heme group. Okay, the heme group is the part that contains the iron. Okay, so if someone is deficient, meaning not have enough in hemoglobin, or if they have a defect in hemoglobin or red cells in general, right? So either you don't have enough hemoglobin, you don't have, or if you don't have enough red cells, or if there's something wrong with the hemoglobin or the red cells, all these cases will lead to anemia. Okay, so there are different types of anemia, but generally, I mean, they vary in severity, but generally you would have trouble distributing oxygen in the body. You would experience shortness of breath, fatigue, and depending on the kind of anemia, it could be painful as well in some cases. Now, if you had me last semester, you probably remember a type of anemia that we constantly talked about, right? Anybody know what I'm referring to? That's right, the sickle cell anemia, right? Very good. So sickle cell anemia is caused by a point mutation in the hemoglobin sequence. Okay, you don't have to memorize what kind of mutation it is, but once you switch that nucleotide, the hemoglobin misfolds, and the misfolding of the hemoglobin causes the red cells to change its shape. So this is a normal looking red cell. It's like a nice disc with a little indentation in the center, again, to maximize surface area. But the sickle cell looks like this, okay? And that shape makes it difficult for the red cells to go past capillaries. They get stuck, and it causes quite a bit of pain. Okay? And not to mention, of course, it's difficult to transport oxygen when they're all stuck in the capillary, right? So that's one type of anemia. Remember, there are different kinds of anemia. Okay? You don't have to know the rest of it, but I'll give you a few examples. If you have iron deficiency, if you don't have enough iron in your bloodstream, then you will not be able to transport oxygen as efficiently because you don't have enough hemoglobin, right? That's iron deficiency anemia, right? So that one won't be painful, right? Like you just have to take iron supplements, right? And then I'll give you another example in a bit. But first, I want to tell you that the sickle cell anemia, because it's caused by a point mutation, it is actually a recessive mutation. You remember dominant and recessive, right? I hope you do. We spend quite a bit of time talking about it. So someone who is like big S, this is normal. Normal RBC. The person is not going to have sickle cell anemia. Little s, they are going to have sickle cell anemia, okay? What about big S, small s? Anemia or no anemia?
No anemia with carrier.
That's right, they're carrier of the trait, but then they are normal, right? They are normal, okay? They're normal. Normal in the sense that they don't have the symptoms of sickle cell anemia, but because they do have one copy of the small s, some of their red cells would have the sickle cell shape, okay? But it's just a subpopulation. It's not enough to cause problems because you also have the normal red cells in the body, right? Do you guys understand that so far? Okay, so the best one to have. Sorry, Hiba, did you have a question or?
Yes, can you repeat this point, please?
Can I explain one more time? Yeah, sure. Okay.
Yes, please.
So big S is normal, but no mutation in the chromosome, and therefore all the red cells are circle and good looking, right? Little s, both chromosomes are mutated, and all the cells are going to be the sickle shell, like the SS shape, right? Okay, and that's bad because they will get stuck in the capillaries and then they won't be able to transport oxygen. So far, so good, Hiba?
Yes, yes.
Now, in the middle, big S, small s means you have one normal copy, which means you are still able to make normal red cells. The little s copy will make a little bit of a sickle cell, but that's okay because you have a good, healthy pool of red cells available. Okay, so that's the situation.
But if
So,
it is recessive, sorry,
yeah,
if it is recessive, how come it has the sickle cell shape? It should not be within the recessive trait.
yeah, because okay, so like the gene, you have two genes, right? Two genes on one on each chromosome, right? The normal S is making normal red cells. It's making enough normal red cells to cover up the copy that's making bad red cells. You're still making the bad one. You're still making the sickle cell one, but then you know, because you have enough good ones that it covers it up, so you don't experience any symptoms. Does that answer your question? It's not like color where it completely covers it, right? I know it's a little bit different compared to like eye colors and stuff like that. Okay, but the point is this, guys. Okay, if you are sickle cell anemic, if you have sickle cells in the body, it turns out you are immune to malaria. Okay, for those of you who don't know, malaria is a disease that is transmitted by mosquitoes. And in developing countries where they don't have access to anti-malarial drugs, people die from malaria quite frequently. Okay, so what happens is, because if you're sickle cell anemic, you cannot get malaria. Then the best genetic makeup to have is actually big S, small s, heterozygous. Okay, because if you're big S, small s, you are both immune to malaria and at the same time not have the symptoms of sickle cell anemia. Right? So if you look at the heat map, the one on the left, the green one, shows where malaria is prevalent. Okay, like parts of the world where there's a lot of malaria cases. And then you look at the right one, that shows you the frequency of big S, small s, like what population, what percent of the population is heterozygous for big S, small s. You see the two heat map actually coincide, right? It overlaps, right? So people in those countries where there's a lot of malaria cases, this is the most protective phenotype to have. Okay. So if you're big S, it's not good to live in those places. You're gonna get infected by the malaria. If you're small s, okay, maybe malaria is not gonna kill you, but then you have sickle cell anemia, which is a debilitating condition to have in general, right? All right, if you have any questions, feel free to type them for me. All right, next slide. Where do we get red cells? From the bone marrow. Okay. In the process called hematopoiesis, right? A couple of slides ago. Now, what happens is when you have a decline in RBC level, right, when you don't have enough red cells, your kidneys actually will release a hormone that helps stimulate red cell production. Okay, so that hormone is called EPO, which stands for erythropoietin, but we can just say EPO. Right? So athletes, they could train at higher altitude. When they go to higher altitude, air is thinner, they have less oxygen in the body, and then the kidney is gonna pump out some EPO and make more red cells. When they come back to sea level, they are more efficient, right? Because now they have more red cells, right? But EPO can actually be manufactured. Remember the biotechnology that we learned last semester? We can make insulin, we can also make EPO, because EPO is a hormone is a protein. If we know the gene for the protein, we can make them. So you can make EPO, and it's being used for cancer patients. Okay? Cancer patients who are receiving chemotherapy or radiation therapy, their bone marrow is being suppressed. Okay, chemotherapy and radiation therapy basically kills off things that are rapidly dividing, right? Like cancer cells. But then the stem cells in the bone marrow, they're also rapidly dividing. So as a side effect, these patients will suffer from anemia. So you give them EPO, right, to boost the red cell production. People who undergo dialysis treatment for kidney problems, they also need EPO. When the red cells goes through the dialysis tubings, right, a lot of them die off, right, and the person gets anemia. So by giving them a boost of EPO, you compensate the loss of red cells. Okay. Next one. Red cells, they live for around 120 days, about 3 months, 4 months, okay? And then when they are at the end of their lifespan, they get trapped in the liver. Okay, liver is right here, right on the right side, and the spleen. Spleen is like, you know, behind your stomach. Okay, there's a tiny organ in the back, and it's like about the size of a fist. That's where the spleen is. They will get trapped there, and they will get eaten by a type of white cells. Okay, a type of white cells, which we'll learn a lot about later. Okay, so don't worry about it if you don't recognize the name at this point. And they will get eaten there. Okay. When you break down the red cells, there are a lot of valuable resources that we want to recuperate. Okay, so the hemoglobin, we're gonna break down the protein. We can reuse some of the amino acid, and we want to recover the iron. Iron is a precious resource in the body. So you want to recuperate them, bring them back to the bone marrow, and then make new ones. Okay? Pay attention to the next part here. Whatever is not broken down or recuperated, it will get converted into what's called bilirubin. I want you to remember this word, okay? Bilirubin. And bilirubin is yellowish in color, and usually we remove it from our urine and feces. That's why the urine and the feces are the color they are. The yellowish, brownish color is partly because of the bilirubin. Okay, and I mean for the feces is also because of the bile from the gallbladder, but you know most of it's the bilirubin. Now we will learn at the end of this this whole lecture that when there is a mismatch between the mother's blood and the baby's blood, that could lead to a lot of red cells dying in the baby. And when there's a lot of red cells dying, there's a lot of bilirubin. The baby cannot get rid of the bilirubin fast enough, and then it starts building up in the skin of the baby. So what color does the baby appears to be in those situations?
Blue?
Yellow.
Yeah, blue is when they don't have
Yellowish?
enough. Yeah, yellowish is the right answer, right? And do we have a name for that condition? Some of the mothers here might have heard about it, or you know, other people as well. What do we call it when a baby is?
Jaundice?
That's right, jaundice. Okay, jaundice. Okay, so we're gonna talk more about it later. Okay, I'll put the word down here. We won't get to it today, but if you want to read a little bit about it before coming back on Wednesday, you know, you could do that. All right, so that's a lot of information for red cells. I strongly recommend you summarizing it, you know, on a piece of paper on your own. Right? Think about where they came from, you know, think about how to make more of it and characteristics of it. Don't worry about the sickle cell stuff. I have no questions on the test for you. That was just like, you know, for interest sake, right? Okay, like I you know, I like to tell you random stuff because it's interesting, right? But not everything is going to be on the test. General rule is, you know, look at the study guide. It tells you what's important, right? So that's all for red cells. You know, which is also why if you donate blood, you cannot donate it again. Like if I donate today, I cannot go back next week to donate again. You gotta wait. Yeah, you gotta wait for your body to make more red cells. Okay. But if you just donate plasma, it's possible to just donate the yellow stuff. then you can go every week. Okay, that part, it's just mostly liquid water anyways, right? Are we good with red?
I have a quick question.
Sure.
Oh, it's kind of like about the plasma, how you said it's mostly just like liquid or water anyway.
Yeah. Mhm.
So if it's mostly just liquid or water anyway, why do we need to donate it to other people? Why can't they just like quickly make their own?
Yeah. So that was a good question, right? I might have like over simplified it. Remember the plasma, yes, it's mostly water, but then it has these important clotting factors, right? So one reason why someone needs plasma is because they don't have enough of these things for whatever reason, and they need to go into surgery, then you give them some plasma, right? But the plasma is useful, as you will see, in reconstituting the blood. Okay, like if someone needs like RBC, you can just give them red cells. But in some cases, they need whole blood. Okay, not just the red cells. Maybe they suffer from like a car accident or something. They're losing a lot of blood. You can't just give them RBC. You need to mix it with plasma. Okay, and as you will see later today, if someone is type A blood, or like type AB blood or something, okay? Like it's actually not okay to take the plasma and the red cell from the same person and mix it together and give it to them, okay? There are different rules when it comes to plasma and RBC donation, okay? So like that's why you have to have all these different plasma available to mix it with the appropriate red cells, and then you give it to the person as whole blood, okay? That's a short version of the answer. Okay. Oh, there's another question here. Blood clots are formed from plasma. No, blood clots are formed from platelets. Platelets, okay? But in the plasma, there are proteins like fibrinogen and prothrombin, which we'll look at again. These proteins help form the blood clot. Okay, so it's not the plasma that's doing the blood clot. It's the proteins in the plasma, some of it anyways, that helps with the blood clot. Okay, and then the platelet also helps. And I'll draw you a nice chart probably in a bit that helps you put everything together. Okay, so that was a question someone was asking in the chat. All right, white cells now. White blood cells, again, fancy name is leukocyte. Leukocyte. Red cell was erythrocytes. This is leukocytes. Leuko means white. They are produced where? Same place. They all came from the same place, bone marrow. Okay. Now, red cells are only found within blood vessels. They're not supposed to come out. But white cells are different. Okay, they can be in the blood vessels, but they can also be found in tissue fluid as well as some organs. Okay, as opposed to just inside blood vessels for RBC. Okay, red cells, they stay in the blood vessels. They don't come out. The only time they come out is if you have a cut. But white cells, as you can see in this picture, they can squeeze themselves out of the blood vessels at the capillaries. Remember, capillaries are leaky, right? So the white cells can change the shape, squeeze through those tiny gaps, and go into the tissue. Sometimes they stay in the tissue. Sometimes they stay in the organ, particularly if it's an organ that is very susceptible to infection. Okay, like they're very vulnerable to infection, like the lungs. We constantly breathe in stuff. A lot of the stuff that we breathe in can actually cause us to become sick. So we can put some white cells in the lungs, just waiting, okay? Waiting for these bad things to come in, and then they're gonna eat them up, right? Do they work all the time? No, obviously not. And when they fail, then we get sick, right? But we put some there, We call them resident white cell because they basically live there, right? So defense against pathogens, bacteria, viruses, parasites. They destroy dead or dying body cells. I already gave you an example. When when red cells gets too old at the end of the 120 days, the white cells will eat them up at the liver and at the spleen. Okay. Oh, I actually didn't know this point was here. It's basically what I wrote here. Red cells are confined to the blood, but white cells can squeeze through pores in the capillary wall. So I'm going to give you a quick note here that shows you the types of white blood cells.
Excuse me, I didn't get why red cell not past the capillary.
Yeah, because if the red cells comes out, then you have internal bleeding, right? If the red cells comes out from the blood vessels and leaked all over the place, then it's essentially bleeding. And we don't want the red cells to be in the tissue, because if they're in the tissue, once they give off the oxygen, there's no way to gain more, right? We need the red cells to stay in the blood vessels so that they could travel back to the lungs and get more oxygen before returning, right? Whereas white cells, it's okay that they exit and then help fight an infection, right? Does that answer your question?
No, I was thinking about what thing prevent to leak out.
Oh, what prevents the
blood.
red cells from coming out,
Yeah,
right? If
yeah.
they're okay, that's a good question. Let me rephrase it and make sure that I am understanding you correctly. So you're thinking the capillaries are leaky. How come only the white can leak out, but not the red? Is that what you were saying?
Yeah,
Okay. So
exactly.
yeah. So let's say the gap is like this, right? The gap is like this. This is an exaggeration, but let's say there's some gaps here like that. Okay. The red cell are these ones. Okay. They cannot go through because they are too big. And the shape of the red cell is more rigid. Like it's you understand rigid? Like you can't really change it, okay? Okay, so it's just that particular shape. Whereas the white cell here, the white cell that looks like this is more flexible. The white cell can change the shape and squeeze through. Okay. Do you understand? Is that better or?
Yeah, thank you.
Okay, so that's the main reason. All right, let's take a look here. And again, I apologize. I know I'm stretching your attention thin. Oh, the guy is actually here. I just heard the bell. Let's take a 10-minute break and then we will continue, okay? 10-minute. Thank you. Thank you. Okay, I'm back a couple minutes earlier. But does anybody have any questions so far? Anything they want to clarify? No, all is well. Okay, good. Let's take the note then. Okay, let's take the note. So here we are gonna learn about the type of white blood cells. So white blood cell. Okay. Now, when people look at white blood cells under the microscope, they have to stain them. I told you, right? Using special stain that makes them purple in color. And if you study their morphology, okay, their appearance, people are able to group them into two categories. Okay, so one category, they call them granulocytes. granulocytes. Okay, so granulocytes are cells that have visible granules. And on the other hand, we have what's called agranulocyte. Okay, and these ones, they have no visible granules. Okay. And you might wonder, hey, what are granules, right? And I'm going to put a star here. Granules are small particles like proteins that takes up the dye. Okay, like when you stain them, when you apply a dye to the slide, for reasons we're not going to go into, some cells really soak up the dye, and then you see these granules under the microscope. We call them granulocytes. And the other ones that don't take up the stains, they don't take up the dyes, we call them agranulocytes. So this is not a functional distinction. Okay, this is just based on appearance, based on what they look like under the microscope. Do you see the granules or do you not see them? If you see them, we call them granulocytes. If you don't see them, then it's agranulocytes. Okay? Now, there are different kinds of granulocytes that I would like you to know. Okay? The first one is called neutrophils. Neutrophils. Okay? Neutrophils are the most abundant white blood cells in the body. It makes up 70% of all your white cells. Okay? Now, a little bit of the math here to make sure that you actually understand what I'm saying. You have your whole blood. That's all of your blood. 1% of the whole blood are going to be your white cells, right? Within that 1%, 70% of it are going to be these neutrophils. Okay? What do? They are phagocytes that remove anything that is not supposed to be there. Okay, so it could be a bacteria, it could be some dying cells, okay? Anything that's not supposed to be there, they're gonna eat them up. Okay, so another term here I want to help you understand, another star, I'll put it here, I guess. Okay, phagocytes are cells that can perform phagocytosis. Okay, remember phagocytosis from last semester? Right? Eating things, right? Cell eating. So if a cell can do phagocytosis, we call it a phagocyte. Okay? So neutrophils just move around in the body, okay? And it mops up anything that's not supposed to be there. Okay? It clears it out before they cause a problem. So that's the first one, neutrophils. Okay? I have a friend, her son was born with low neutrophil, so low that he cannot go to daycare. He got very sick very easily. But it's something that apparently he was able to grow out of. Okay, once he got older, the numbers rebound, right? And he has a normal immune system. Okay, otherwise, the person would have what's called neutropenia. Okay, not enough neutrophils, so they are essential part of general surveillance defense for the body. Okay? So they are granulocytes as well as phagocytes. Okay, like you can put cells in different categories based on how they look and based on what they do, right? So based on how they look, we call them granulocytes because they have granules. Based on what they do, we call them phagocytes because they go around and eat stuff. Okay, we have another one here. It is called eosinophil. Okay, eosinophils. Sometimes the pathogens are too big for the cells to eat it. If you think about having like a worm infection, and like a worm that grows in your body, right? It's so big that you can see it with your eyes, right? How can a microscopic cell eat the worm? You can't. Right? So that's when you're gonna need some eosinophils. Okay, so they attack parasites. Okay, by secreting chemicals. All right. They do other things as well, but like I just want to give you like a simple tagline, right, for you to remember. Okay. So normal tiny stuff, you can eat them up with neutrophils, no problem. If they're really big, you're gonna need some eosinophil. Number 3, we have basophil. Basophil. They are responsible for creating inflammation. Now, inflammation sounds like a bad thing, but it's actually a defensive strategy your body uses to limit the spread of the infection. Okay. So if you have a cut in the finger, it's red, it's painful, it's throbbing and it's swollen, all these things, that's caused by inflammation, right? And basophil plays a role in that. Okay, when you have allergies, that's another kind of inflammation. Okay, so these are 3 kinds of granulocytes. Does anybody have any questions before I talk about the granulocytes? Sorry, the agranulocytes. Okay,
How
so
can we
sorry.
find out if somebody is deficient in any one of them?
How can you find out if someone is deficient in any of them? Is that what you're asking?
Yes, please.
Yeah, that's my wife's job. Okay, she routinely look at people's sample, and she have to count them. Okay, and found out if they fall within the right range. Okay, so they have to know. She has to know what a neutrophil looks like. She has to know what an eosinophil looks like, and then she have to count the slides and make sure that they fall within acceptable range. Okay, and if it's outside of certain range, then for example, if someone has a lot of basophil, right? That's what she was checking for me at the time, right? With the the picture of my blood, right? She wanted to see if there's a lot of basophil, right? Like if I'm undergoing some kind of inflammation, right? And that would show. If the neutrophil is too low, fall below the acceptable range, then we know they don't have enough. It's physical counting, basically. All right. But with AI and stuff like that, they are actually training machines to count it. And then, you have a human to verify the results in some cases. Okay, agranulocytes. They don't have visible granules. Okay, for reasons we're not gonna go into. They don't take up the dye. Okay, so you don't see these granules. And we have what's called a monocyte. Okay, so monocyte is kind of like, you know, we can call them inactive. Okay, they're not in fighting mode. Okay, they're just like in surveillance mode. Okay, they just move around the body and check to see if things are okay. If there are no foreign bacteria, if there is no, you know, bad things in the body, then they move on to the next site. Think of like like security in a building. They just walk around and make sure everything is okay. And if it's okay, then it's okay. Like you move on to the next part, right? However, if you encounter something that is not supposed to be in the body, they could change, they could transform, they can differentiate. That's the right word. Okay, differentiate just means transform when we use this word to describe cells. Transform into macrophage. This is the active form. Okay, the macrophage is ready to fight. Okay, they will eat up the bacteria. They could eat like up to hundreds of bacteria before they die. Okay, and then they fight off the infection for you. Before the break, I told you some white cells can stay in certain organs because those organs are susceptible to infection, right? They are vulnerable. So like in the lungs. So the monocytes might move to the lungs and then they transform in the macrophage, right? You don't want to wait until something is bad to transform, right? Let's transform first because you know something is going to be coming in, right? Let's get ready to fight. Okay, so this is a one-way transformation. They fight until they die. Okay, once they become a macrophage, there's no coming back to monocyte. Okay, it's a permanent change. All right, so that's that. And then we have lymphocyte. Lymphocytes. Okay, the lymphocytes come from the lymphatic system, which we will talk about later on. This lecture is called blood and lymphatic system, right? So lymphatic system is going to be at the end. For now, I just want you to know that there are two types of lymphocytes. There are T cells and B cells. We're going to spend, you know, a lot of time talking about these in lecture 3. But again, for now, as a summary sheet, T cells, they help coordinate immune attack. Okay? The T cells will be like, hey, you over there, you go eat that thing. Okay? You go make some antibodies. Right? So they're like coordinating the immune attack. And B cells, they make antibodies. Remember antibodies? They are in your plasma, right? So if you have fought off COVID, then in your bloodstream right now, you have some anti-COVID antibodies. They're going to be there for, you know, virtually the rest of your life. So next time you get COVID again, you might not get as sick. Okay. And then I know some of you are going to ask, you know, I got it twice and then the second time was just as bad. That's probably because you got infected by a different variant, right? And we'll talk about all those in lecture 3 as well. But these are the summary for you, okay? Now, as we progress through the course, I encourage you to dig back these notes, right? And then you add details to it, right? Sometimes you might want to rewrite them, right? You might look at these slides again and you're like, oh, we learn more about it later on. Let's add more details to it. But this sheet is certainly something good to have on the test with you, right? Like on the side as a reference. Right? If you have a hard time memorizing them, just have a quick reference sheet is a good idea. For those of you who don't know, right? Like I have been telling the class, it's okay for my class to use notes. Okay, I encourage you to use notes during the test. Right? Write a summary sheet. Okay, it will help you do better. So that's it for the note. Does anybody have any questions for me? Please, you guys are awfully quiet. No, everybody's happy? Okay, that's fine. Let's go back to our PowerPoint here. That's what they look like. You don't have to identify them. I just put it here in case you are interested to see what they are. Okay, so like if you look under a microscope and you see this guy with the 3 balls, you're like, oh, that's the neutrophil. Okay, and that's what an eosinophil looks like, and so on and so forth, right? So the ones on the left here, those are the granulocytes, and then the stuff on the right, those are the agranulocytes, right? We only talk about these types, okay? Again, later on, we might add a few more. So this is just for your reference only, okay? Don't go memorizing what they look like. It takes a lot of training, right? I don't even recognize them. Okay, that's it for red cells. Red cells are erythrocytes. That's it for white cells, leukocytes. And now we have the last type. Okay? The last type is called platelets. Okay? And everybody has to have a fancy name, okay? It's not my fault. I don't know. Whoever created these, they're like, let's give them difficult names. So the fancy name for platelets, it's called thrombocytes. Okay, thrombocytes. Now, interestingly, the platelets are not cells per se. They are just fragments of a cell that has broken off from a bigger cell. That bigger cell is called a megakaryocyte. That's this thing right here in the center. Mega means really big, right? Megakaryocyte. And all these little fragments that are breaking off, these little red bits and pieces, that's the platelets. You make so many platelets every day, okay? 200 billion platelets per day. Right? They don't have nucleus, right? They lack most cellular components, and they only last for about 10 days unless you use it beforehand. Okay? And what do we use platelets for? To stop the bleeding, right? Stopping the bleeding is called hemostasis. Do not confuse hemostasis with homeostasis. Okay, homeostasis is what we learned before, right? Maintaining internal balance. Hemostasis. Hemo means blood. Stasis means to, keep it at a certain level. So this is to stop you from bleeding. Okay. Stop the bleed. Okay. Let's take a quick note on coagulation. Coagulation is another word for blood clot, okay? There's so many words for blood clot, right? Coagulation, hemostasis, blood clot, right? All right, so here we go. All right. So here is a blood vessel. And the blood is going by no problem because the inside is smooth, right? It has these smooth endothelial tissue, no rough patches, blood just goes by without stopping. And then you cut yourself with a thumbtack or something. Once you have the injury, you're going to be exposing some of the underlying tissue. Okay? The best way I can think of, like as an analogy, is like think of carpet, okay? Like you walk on the carpet, it feels nice, it's fluffy and smooth. But if you scrub it out and pull out a chunk, then all the yucky stuff from below is exposed, right? Like you will see the dirt, you will see the the foamy stuff and whatever is below, right? That's kind of like it here. So when you have an injury like a thumbtack, it exposes the underlying tissue. Okay? So the damaged tissue releases, I don't know why I'm writing in all caps, damaged tissue releases what is called thromboplastin. Okay? These are not supposed to be in the blood regularly. It's only supposed to be in the blood when you have an injury in the blood vessel. Okay, so there's a thromboplastin that's coming out right from here, right? All these thromboplastin now. So earlier today, I told you in the blood plasma, we have these coagulation proteins, right? We have the prothrombin, we have the fibrinogen, right? So let's write down prothrombin here. Prothrombin. Now, prothrombin, the pro means it is inactive. This is inactive. You need to turn it on, okay? You need to activate this thing. Okay? So, in the presence of thromboplastin, the prothrombin will get activated. And to do this, you need a little bit of calcium as well. Okay? And then it becomes thrombin. So this is active. Okay? So right now in your body, assuming you don't have any cuts anywhere, you have these prothrombin just circulating in the bloodstream, in the plasma. It's going around the body. But anytime you have let's say you have a cut here or like a cut like wherever, the cut is going to be releasing the thromboplastin, which will convert the prothrombin into its active form, the thrombin. Do we have any questions so far with this before I go on to the next step?
So first the prothrombin is released and then the thrombin is released.
So prothrombin is already in the blood the whole time, but it's just not turned on because there's no reason to turn it on unless you're bleeding, right? Turning on the prothrombin will lead to formation of a blood clot. We only need a blood clot when we have a cut, right? Okay, so prothrombin is not released. Right? This stuff is already there in the blood right now. This stuff is released. Oops. That stuff is released. Right? That stuff, it was like all covered up, nicely tucked beneath your vessel connective tissue. Once you damage it, then it comes out. Does that answer your question?
Yeah, so saying like the cut turns on the prothrombin and the thromboplastin is released, is that correct?
That's right. So the cut. So first there's the cut, then the thromboplastin is released, and the thromboplastin will turn on the prothrombin. Okay.
I have a question.
Yes.
So since you're talking about bleeding, is it only when you have a cut or it applies to monthly menstrual flow of women?
Oh, so like menstrual cycle is the shedding of the blood vessel, right? So like there is some damage of the blood vessel in that sense. That's why like the menses sometimes might have a little bit of blood clot in it as well. But this process applies to all kind of vessels damage. Does that answer your question?
Yes.
Okay.
Thank you.
All right. So what's the other one? Right? If you remember, it was called fibrinogen. Fibrinogen. And ogen means it is inactive. So there are some prefix like pro and suffix like ogen. They mean it is inactive. And to turn on fibrinogen, you're going to need the thrombin. Okay, and then it becomes fibrin, which is the active form. Okay, we're almost there, right, with this note. Okay, I know it's like, it looks like it's like so many layers. But the basic concept is one thing leads to the next. It's like dominoes, right? Dominoes, like those little tiles. Like you push one, and then it causes the next one to fall, and then it causes the next one to fall. So these are called reaction cascades. They are basically like dominoes. Okay? And that's it. That's it. Once you form the what is fibrin? Fibrin is a protein that can form these strands. Okay? So they form these big strands, okay, around the damaged vessel. Okay? And the strand becomes a net. What does the net do? The net will trap some red cells. And then, more importantly, it will also trap some platelets. Okay, and eventually, when you trap enough of these red cells and platelets, it seals the cut, right? And you have the scab, right? As long as you don't peel the scab, you're not gonna bleed again. So that's what this is for. Okay, this is one way to clot the blood. There are other more complicated pathways which we're not gonna look at. I'm just telling you because in case you try to Google it online and then you see something completely different, and you're like, why are we not learning about this, right? Like we're trying to keep it simple, right? And as you can see, even trying to keep it simple, it has a lot of different steps. Okay, but I want to show you the picture again in the PowerPoint. Look, that's what it looks like for real under the microscope. The white strands, that's the fibrin forming the net. And that net is important to catch all these red cells, and eventually it kind of paves the hole. All right? And then you stop the bleeding.
I have a quick question about the note.
Let me go back.
So once you get a cut, the damaged tissue releases the thromboplastin, and then the thromboplastin turns on the prothrombin.
Yeah, prothrombin. Yeah.
And then where's the thrombin like happening in there? I'm a bit confused with that.
Okay. So the prothrombin in the blood will become the thrombin. Okay. It doesn't go anywhere. It's just like prothrombin, and then you activate it, and now it becomes thrombin, and it's still right there. But then now the thrombin will go grab onto like a fibrinogen and change it into fibrin. Does that?
Okay, so then after it transforms into the thrombin, it goes and like attaches itself to the
fibrinogen
fibrogen thing.
Mm-hmm.
And goes over to the other one and creates the scab.
That's right. Which creates the strands, and then the strands trap all these things, and then it becomes the scab, right?
Okay, thank you.
Yeah. So remember, I want to reiterate that in our body right now, we have the prothrombin and we have the fibrinogen. They are in the plasma. They are just circulating. Okay. And then when you have the cut, that's when you have to activate them. Understand? This also needs calcium, by the way. That's all, okay? I think there is a question on the test where I list the steps and I ask you to rearrange it. Okay, that's the only question I'm going to ask you. Okay, so look at the chart. Right? Thromboplastin gets released. Prothrombin gets converted into thrombin. Fibrinogen gets converted into fibrin. Is all. Okay. This is a summary of everything we talked about for the past hour and whatever, 15 minutes. No, more than that now, right? Almost 2 hours now. And you can read it. Okay. Obviously, don't memorize the numbers. Okay. But that's everything. Okay, so that's the boring stuff, guys. Okay. Oh, sorry, not yet. We still have to do some poll questions, and then I'm gonna get onto the exciting stuff, the genetics. Okay, that's really fun. But let's do some polls. Okay. Again, please try to participate if you are able to. Okay, most of you got it. Thank you for participating. You just have to look at the note that we took, right? And that is the basophil. Okay, basophil is a granulocyte. It releases chemicals to cause inflammation. Next question. Anyone else? Okay, thank you. The correct answer is parasitic infection. Okay, the parasites are too big. You cannot eat them with neutrophils. You cannot eat them with like macrophage. So you're gonna have to use chemical to attack them. And that would be the eosinophil. Eosinophil is the second one in the notes. Okay. This one usually gets people a little bit confused. Okay, last call. Thank you. Hemoglobin is found in red blood cells. Okay, red blood cells. Right, that's the protein that carries oxygen, right? And carbon dioxide actually as well. And it contains iron. Okay, that's it. That's it for the background information on blood. Okay, so just to quickly recap, we talk about the plasma and everything that's in it, right? The proteins specifically, right? And then we talk about the different types of formed elements, the red, the white, and the platelets. Okay, and within the white cells, there are like so many different kinds, right? All right, let's move on to the genetics. Okay, like I said, this is very interesting. I found it very interesting, but it's also a little bit challenging. Okay, because there are some word problems that you have to solve. It's kind of like the genetic unit that we did, last semester, right? Okay, first I want to ask the class, do you know what your blood type is? Anybody want to share their blood type? I mean, you can make it up, like I would never know.
O plus.
O plus, okay, and then I have people
A
say
plus.
B plus. Okay, very good, A plus, yeah. So when you describe your blood type to me, you actually told me two things. You tell me whether you're A, you're B, or O, or AB. That's one blood group. And then you tell me you are plus. Okay, so plus or minus, that's actually an other blood group. Okay, but did you know in the human body, those are not the only blood group. On this picture, it shows you some of the common blood group. So when you tell me you are AB or ABO, that's the ABO blood group. When you're saying whether you're positive or not, you are talking about the RH blood group. Those are the common ones that people usually know, if they know at all. But then there are other ones that needs to be taken into consideration when we do a blood transfusion. Okay, we have things like called Cal, Indian, Chroma, Duffy. All these are blood types that exist in the body. And if we want to do a safe transfusion, and we have time to do the testing, we actually have to consider all the different types of blood group. So you might wonder, what exactly is blood group? Well, blood group or blood type refers to the inherited antigens present on the surface of a red blood cell. Okay, so what is inherited? Inherited means you don't get to choose your blood type, okay? You're born with it. It depends what genetic material you inherit from your parents. And antigens, okay, you want to write this down. Antigens are basically molecules on the surface of a cell. Okay, it could be surface of a bacterial cell, it could be surface of like your normal cells, but in this context, we are talking about the surface of your red blood cells. Okay, so what kind of molecules do you have on the surface of your red cells? We call that the blood type. Okay, so when you say you are type A blood, then you have the type A antigen on your red cells. Okay, so they could be proteins, they could be carbohydrates, sugary proteins, or sugary lipids. Okay, and right now we have discovered a total of 40 different blood groups. Okay, this is a link to Wikipedia. You can click on it and we show you all the different types. Okay, I'm not going to do that. You can do that on your own. According to the Canadian Blood Service, most Canadians are O positive. Okay, followed by A positive, and then it's kind of rare to be AB negative. Okay, I am AB positive, okay, so I guess I'm kind of rare too on this scale. But you know, it's a diverse blood type. So the ABO blood type, that's what we're going to be focusing on for probably the rest of today's class. Next class, we'll talk about RH as well. Okay, but we're not going to learn all the other ones. Okay, it's like too much. So ABO is essentially based on the presence or absence of antigens, antigen A and antigen B. If you have antigen A, then you are type A blood. If you have antigen B, then you're type B blood. If you have both, then what type of blood are you? AB, right? Yeah, exactly, AB. Right? What if you don't have any? If you don't have A and you don't have B, what's the blood type?
O.
O. That's all. There are only 4 possibilities. A, B, AB, or O. Okay. So the alleles that are responsible for forming the ABO blood group are listed here. Okay. These are the official symbols we use. It's either IA, IB, or little I. Now, we are diploid organisms, right? We have 2 sets of chromosomes. So even though there are 3 alleles in the population, at most, we can only have 2. Right? At most, we can only have 2. So let's take a note on this blood type stuff. And then we'll do some problems. Blood ABO blood group. So let's do a little table here. Actually, if you printed the study guide, we have this table there as well. But you know what? It's probably just easier to keep everything on the same page here. Okay, so this is blood type. Blood type. Okay, and then we have genotype. Okay, and then we'll put RBC here. And then we will add some more later on, okay? For now, just gonna have 3 columns. I'll give you a moment to draw that. Okay. We have type A blood here, type B blood, AB, and O. 4 types, right, in the ABO system. Now, genotype. So, if you are type A, your genotype, as in your genetic makeup, may be IAIA. So you are homozygous IAIA, or IA little I. Okay. If you have the IA allele, you can make the A antigen, and then you would have it on the surface of your red cell. Little I doesn't make anything. Okay. But you don't need to have 2 copies to make antigen A, right? You just need 1 copy, right? Again, loose analogy is like this. Let's say I have cookie cutters, okay? If I have 2 star-shaped cookie cutters, I can make star cookies. Okay? But if I only have 1 star cookie cutters, that's okay. I can still make star cookies, right? Okay, as long as I have 1 star shaped cookie cutters, I can make star cookies. Same situation here. As long as I have 1 copy of IA, then I can make A antigens. Right? It doesn't matter if I'm homozygous or not. If I just have 1 copy, then I can make it. So your RBC, okay, I want you to use colors here. Okay. On the RBC, we're gonna have A antigen. Okay, remember, we're gonna add more stuff to this chart later. Can anybody tell me what the genotype for type B would be?
IB, IB,
IB.
or IB, small I.
That's right. Okay, both of you are correct. IB or IB small I, right? And for the red cell, please use colors, okay? It will help you understand more later. You will have the B antigen. Okay, remember, these are just like, proteins, right? Or carbohydrates. We're not gonna distinguish them, okay? This is just molecules on the surface. What about AB? What's the genotype for AB?
Only IA and IB.
IAIB, that's good. Now, in this case, both of them will show up, right? Both of them will show up. Does anybody remember what this is called in genetics? If both alleles shows up. Good try. A heterozygous just means the 2 alleles are different. For example, this is heterozygous, this is heterozygous, this is also heterozygous. But if they both show up, right? Then anybody remember? You're almost there. You're missing a little 2 letters at the front there. Anyone? No? Oh, there you go. Very good. Okay, codominance. That's right. This is codominance. Okay. So actually, I'm gonna put it at the bottom here. IA and IB are dominant over little I. That means it covers it up, but are codominant with each other. Huh? Okay. So when they are together, they will both show up. Not a blend, right? But they both show up. But if they are with little I, it covers it up. Okay? So how can you be type O? Type O is no A and no B. So that's just little I, little I. That is all. Any questions about the chart before we do some genetics problem? No? Okay. Let's do this. A man with AB blood is married to a woman with AB blood. What blood types will their children be, and in what proportion? Okay, so the man here, the man is AB blood. There's only one way to be AB blood, right? IAIB. And the woman also IAIB. Okay, I hope that makes sense. Look at the table, look at the chart. So the man is gonna make sperms, and during sperm production, the chromosome separates. Half of his sperm will have the IA, the other half will have the IB. Same as the woman for eggs. Half of her eggs will carry the IA, the other half will carry the IB. You remember how to do this Punnett square, right? I surely hope you do.
Yeah.
So you put them together. It's okay if you don't remember this, okay? Just like message me, email me, and I can meet with you to help you, okay? It's not a problem at all. So there is a 25% chance their kids is going to be type A, right? Right there. There is a 50% chance that their kid is going to be type AB. And 25% chance is going to be type B. That's all, right? Any question? Simple enough, right? Okay, but buckle your seatbelts. Things are about to get crazy. Suppose two newborn babies were accidentally mixed in the hospital, and there was a question of which baby belonged to which parents. From the following blood types, determine which baby belongs to which parents. Now, I want to tell you that's not what they do in real life, okay? You cannot determine if they are your own kids or not just by looking at blood type. You know, we have to do DNA fingerprinting, right? But this is just for, you know, just for exercise purpose, right? Actually, quick story. When we had our first baby, one of my wife's fear is that we would bring home the wrong baby. Okay, so she gave me a mission. She's like, okay, when the baby is born, okay, your job is to stay with the baby wherever the baby go. Okay, and then I was like, but what about you? Like you be, you know, bleeding and all, and you know, I wanted to be there for you. She's like, no, don't worry about me. Okay, just stay with the baby and make sure that you know she's not gonna get swapped or something like that. Okay, so it's like, okay, fine. Right? But it turns out in Canada, the baby never leaves the mother. Okay, that's not the case, right? In Hong Kong, for example, that's where I'm from, and because her sister gave birth in Hong Kong, and that's why she would worry that the baby's gonna leave the room, and I had to follow her, right? But you know, no, the baby never left a room. And you know, I'm pretty sure we bring home the right baby because she looks exactly like me, according to everyone that met her. So it's kind of unfortunate for her, I suppose. But back to the question, we have the you know, blood type of the babies and the blood type of the parents. Can we figure out which baby belongs to which parent just by looking at the blood type? I'm gonna give you you know, a couple minutes to figure it out. You shouldn't need to do a Punnett square, okay? You should just be able to
AB1
logic.
belongs to type O, belongs to the second type B, and baby 2 belongs to type A, B, and type B. Mr. James, and baby 1 goes to Marshall.
Okay, so that's what Mohammed is suggesting. Someone else, Tahib, you were saying something as well?
Yes, baby one belongs to Mrs. Machau and Mr. Machau.
Okay,
And then the second one belongs to Mrs. James and Mr. James.
wonderful. Let's give the class a few more minutes to try, maybe another minute to see if they agree or if there are other suggestions. And then I will tell you some secret about how to do these questions. All right. So what you should be thinking about in a situation like this is, you know, the genotype of the babies. Okay? Let's take a look at baby O, baby 1. Okay, baby 1 is type O. So according to the chart that we wrote down, there's only one possible genotype, right? Okay, and that is going to be little I. Okay, which means, listen carefully, which means if a person is type O, they must inherit a little I from mom and from dad, right? Okay, one little I from each parent. Let's take a look at the James. Okay. The father is type AB. That means the father is IAIB, which means it is impossible for an AB person to have a type O child, regardless of who they marry. Okay, let me say that one more time. Okay, I'll write it out for you in a bit. Okay, but listen, it's impossible for an AB person to have a type O child, regardless of who they marry. Okay, because to have a type O child, you need to pass on a little I. You don't have a little I to pass on when you are AB. Does that make sense? Based on that alone, we can conclude baby 1 belongs to the Marshalls. Okay, and then if there are only 2 babies that got mixed up, thank goodness, then you know baby 2 belongs to them. Okay, so AB person, okay, and a AB person cannot have O babies. Doesn't matter who they marry. Okay, you can marry a type O person, not gonna matter. Similarly, can you give me another sentence? Like another statement that's always true along this context? Anybody?
They cannot donate blood to old people.
Okay, thank you for bringing up donation, but we're not there yet. I'll come back to you when we do get there. But in terms of like what kind of babies you can have and not have, is there another restriction here? So we know AB cannot have O babies. Anything else? Like another statement maybe?
The O type cannot have A, B, or A. If it's supposed to.
Okay, I agree with the first part there. Okay, so let's write that down. O person, like a type O person, cannot have AB babies. Okay? To have AB babies, you need to contribute IA or IB. Your spouse would do the other one, right? But if you are type O, you can only pass on little I. You cannot have an AB baby. Okay? I need you to understand these 2 statements. It will save you a lot of time on the test. Okay? I'll give you some practice example later. Right? Now, some of you might be wondering, right? Some of you might be wondering, how is it possible for these people to have a type O baby? Right? I mean, they're both type B. Where does the little I came from? Look at the table. Look at the chart. Where is the chart? Right here. If you are type B, you can be homozygous, IBIB, or you could be heterozygous, right? So if they are both, in fact, they have to be both heterozygous. And if you do the Punnett square, there's a 25% chance they will have a type O baby. Blessing, do you have a question? I see that you have a mute.
Lucky me.
Oh, yes, sure. Go ahead, please.
I am O. My wife is B. My son is B.
That's okay. Yeah. So like you said you're O, right? So
Yeah.
little I. Your wife could be IB, or
Yeah.
IB little I. And then if you do the Punnett square, like if this is the case, then it's 50% chance. Oh, sorry. In this case, it's 100% chance. 100% chance B, right?
100%.
That's
B.
right. That's right. But if your wife is IB little I, then 50% chance, right? Okay. Now, there are some, exceptions here, okay? Like one semester, one person came up to me at after class, was like, "Oh, I am AB, but my daughter, like biological daughter, is type O. Like, what's going on?" All right. So like there are exceptions, but we're not going to go into the exceptions, okay? All I'm saying is, don't go and file a divorce based on what you learned today, okay? There are always exceptions in biology with these rules.
Question.
Mm-hmm.
So how would we determine Mrs. James's blood type? Would she be like IB or IBI?
Oh, yeah. I mean, like in this case, that's a great question. In this case, we don't know. We don't know for certain. Okay. And in some cases, you have to look at more kids. Okay. So like take the example we just did, right, with the student. He was saying that his wife is IB. We don't know. He just said his wife is type B, right? So how do I know if they are IBIB or IB little I? Well, let's say they have another child that's type O, right? Then immediately we know it's heterozygous, right? Right. But like if they have like 3 or 4 kids and they're all type B, then we cannot say with certainty, but it's more likely is going to be IBIB, right? Does that make sense?
Okay, yeah. And then we got baby one is the Marshall baby because for the baby to be the James baby would be impossible because of the AB
That's
can't
right.
have.
And by process of
Okay.
elimination, we know it has to be the Marshalls, right?
Okay.
Okay, you
Thank
guys
you.
gotta. I'm sorry. Go ahead. Oh, you just say thank you. Okay, yeah, no problem.
Yeah, I just said
So like we're gonna learn more about this next class, and we're gonna have like 4 babies and 4 parents, okay, and more blood groups. So please understand this, and yeah, do your homework, okay? All right, we have the Shrek family here, and this is a pedigree basically. Circle means female, right? Square means male. Sideways means connected to get married and have kids, okay? So your job is to figure out everybody's blood type and genotype, and Fiona specifically, okay? We don't have anything about Fiona, okay? I'll give you a moment. This is gonna be the last thing we do today, okay? So if I'm straining your attention, don't worry, relief is on the way. Let's just finish this one. Okay. I'm just gonna put it in the note here. There we go. That's a good question. Okay, we have some answers already. Yeah, feel free to share your answers on the chat. if you have it. Okay, you guys are saying A, right? But I want to know more. Is it like homozygous A, heterozygous A? You know, what's going on with these people?
Fiona will be IA small I.
Yeah, okay.
Type O will be II. Type B will be IB small I. And type A will be capital IA small I. Queen will be IA small I.
Okay, so you got it all figured out, right? Very good. That's correct, by the way. But let's take it out. Let's take it up, okay? Let's take it up and see how you would do this on the test. And you will have some of these questions, okay? So the way to approach this is to write out the genotype of people that you are certain of, okay? So like King is IAIB. Fargo here. I look it up on Wikipedia. Those are the actual names of the kids, apparently. Little I here. And that's all we have, okay? Those are the only individuals we know the genotype of. The rest, we know half of it. Right? We know half of it. Okay. Then we gotta do some detective work here. Okay. Fargo is little I. So one of the little I's must have came from the father, right? Shrek. And the other little I must have came from Fiona, right? Where did Fiona get her little I? Well, not from the king.
Green.
Must be from the queen, exactly, right? So that's, you have to trace through the generations, right? All right, let's take a look at Fergus here. Fergus has an IA, but the IA could not have came from Shrek. Shrek is type B, right? So it has to come from Fiona. And in fact, if you trace it back another generation, you know Fiona got her IA from the king, right? Okay. What about the other allele for Fergus? The other allele has to come from Shrek, right? Because he already got the IA from the mom. The other one has to come from the dad. It cannot be the B because that would change the blood type to AB, right? It must be the I. For Felicia, the IB must have came from Shrek. And the other one must be the little I from the mom. Any questions?
No, that's
Could you explain one more time?
From the whole thing or which part on?
Yeah, please, whole thing.
Okay, sure. So we start with the ones that we are certain of. So I want to ask you, Hong Yong, which blood type do we know for certain about the genotype?
O.
Okay, so O, we always know O is little I, right?
Yeah.
And what's the other one?
B, capital I, little I.
But we don't know, right? Because B can be capital, could be IB as well, right?
Yeah.
Okay. Now, for anyone who understand this already, please try the next two concept track question. Okay, in the PowerPoint slides. Okay, I'll take them up shortly. AB would be IAIB, right? Those are the only ones we know for certain. Is that okay? right? Those are the only ones we know for certain. Now look at the chart, right? For these ones, A and B, we don't know if it's homozygous or heterozygous. We have to investigate, right? So that's why we started with writing down the king and for this baby right here. Okay, so let me just erase the rest of it for now. Okay, so now I want to ask you, look at this guy. This guy is little I, right? He must have inherited one from each parent. And that's how I know there is a little I here and a little I here. You still with me?
Yeah, I got it.
Okay, so now look at Shrek. He is type B. So what is type B? Look at the table. The table B
Capital.
can be, yeah, IB or IB little I, right?
Yeah.
So we know it has to be like this. Make sense?
Yeah.
Okay. So then over here, type A. Type A is IA something. Type B is IB something. Again, I don't know at this point whether it's IA or IA small I, right?
Yeah.
Okay, so then we do some investigation. Okay, where would the A come from? Well, the A over here, the A has to come from Fiona, because this guy doesn't have an A, right?
Yeah.
That's right. So what can be inherited? It cannot be the B because it will change the blood type to AB, right?
Yeah.
That's why it has to be small I. Okay?
Yeah.
And so on and so forth. All right. So maybe you want to rewatch that recording at the end a little bit, see if you get it. Otherwise, we can talk about it more again. Okay.
Yeah.
All right. Let's take up these questions. Okay. Anybody? I'm not gonna do the poll. Okay. I'm just gonna ask. Anybody have an answer for me? For John.
It's IBI.
IB, like this one?
Yeah.
Yeah. Okay. I have no idea. I gotta do it. Okay. So we should start with Sue, right? Sue is little I. Mary is IAIB. Harriet is IA something. But actually, I know it has to be IA little I, right? Has to be because then Ed, her father, is little I. So she must inherited one little I from Ed, right? So let's take a look at John here. Okay, what's John? Well, John and Harriet has a child, Sue, that's little I. So one of the little I got it's inherited from Harriet. The other one must be from John. Okay, so John has one little I for sure. And what about the other one? Well, the Mary, the like their kid Mary, IA came from Harriet. The IB must have came from John. There's no other way. That's why the answer is B. Okay?
Yeah.
Last question. What is the blood genotype of Mona? Okay. Anybody got Mona? It's the same setup, right, as last time. So we know Ed is little I, IA little I, IB little I. These ones don't matter anymore. What's the genotype of Mona? A? Yeah. Anyone else?
Must be A.
Must be A. Yeah. So this is a trick question, okay? Because a lot of people, for some reason, myself included, sometimes look at this and they thought that they came from the same parents, but that's not true, right? John is not related. Okay, siblings don't get married, right? Not supposed to anyway. So like the only thing you're looking at is this. Okay, the John's blood type has nothing to do with Mona's blood type, right? Okay. So, what is Harriet's blood type? IA little I. The little I came from Ed. and the IA came from Mona. But there is no way for us to know whether it is IA, IB, IA, IA, or IA small I, right? We don't know, right? Could be IA, 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 IB, or IA little I. We don't know. We don't have enough information. Remember, John is not in the picture. He's not part of the, biological family, right? Need more information.
Yeah. If we don't E, there not is E. Option A, it was correct.
No, we don't know. We don't know for certain if she's IAIB. She could as well be the other ones as well, right? All three are possible.
Occur. One probably occur.
That's right. One probable outcome exactly. But it says here for sure, right? That's why it's wrong, right?
Yeah, you're right.
Okay.
Thank you.
Yeah. All right. So we learned a lot of stuff. We still have a lot to learn on Wednesday. I will post the information about the quiz as soon as I can. And again, if you need extra help, please do not hesitate to reach out to me. We can set up some time to meet on Friday online, and we can work these things out. Okay. Thank you for coming. Okay. See you on Wednesday and stay warm. Bye for now.